Welcome back, everyone, to today's Explainer. Whenever we hear the term transgenic animals in the news, it's super easy for our minds to immediately jump straight to like a crazy science fiction movie, right? But the reality is actually much more grounded, and honestly, it's way more fascinating. These creatures aren't some wild sci-fi experiments. They are the fundamental biological architects that are literally driving modern medicine forward. So today, we're going to look really closely at the science behind transgenic animals and explore how they're carefully crafted to help us understand, treat, and ultimately conquer human disease.
Okay, let's dive into this and take a quick look at our roadmap for today. First up, we'll officially meet the transgenic animals. Second, we're going to see how they help us study human physiology and disease. Then, we'll look at their incredible role as living pharmaceutical factories. And finally, we'll wrap up with how they're used for testing safety and toxicity.
All right, Section 1. Meet the transgenic animals, the absolute foundations of biotechnology. To really grasp the applications we're about to see, we first need to ground ourselves with a clear definition. Basically, a transgenic animal is an animal crafted through the direct manipulation of its DNA so that it carries and expresses additional genes, which are often from entirely different species. By introducing these foreign genes, scientists are fundamentally rewriting the organism's genetic blueprint. It's a highly precise molecular innovation, and it's exactly the engine propelling some of our most groundbreaking medical advancements today.
Now, when you hear genetic engineering, you might picture some exotic creatures in a top-secret facility, but the reality of the lab is far more grounded. And this brilliantly illustrates exactly what I mean. Look at this. The vast, overwhelming majority of transgenic models used in science today, we're talking over 95%, are literally just mice. Only a tiny 5% makes up all other species combined. It just goes to show that the humble laboratory mouse is absolutely the unsung hero of modern biotechnology. Let's move to and see how this builds into Section 2, studying physiology and disease, basically unlocking biological mysteries. How do researchers actually use these little guys to decode our own human bodies?
Well, a primary objective here is to map out exactly how bodies grow and function, and the sequence they use is actually pretty elegant. Step 1, you introduce carefully selected foreign genes. Step 2, you study the regulation of those genes, like when do they turn on and what turns them off. And then step 3, you observe the tangible effects on bodily functions. For example, if scientists manipulate an insulin-like growth factor in a transgenic mouse, they can literally watch the cause and effect happen right there on a genetic level to decode physical growth and development. It's really amazing.
But of course, the application goes way beyond just normal physiology. We're talking about modeling severe human diseases here, things like cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. By engineering these mice to carry the exact genetic anomalies associated with these human conditions, researchers essentially create active, living mimics of the disease state. This means they can understand exactly how genes contribute to disease progression and actually test potential life-saving treatments way before they ever reach human trials.
Which brings us to Section 3, living pharmaceutical factories, engineering biological products.
So we know the pharmaceutical industry relies heavily on complex biological products to treat illnesses, right? But the catch is, producing them is usually incredibly expensive. So scientists thought, what if an animal itself could manufacture the medicine? Well, enter Rosie. Born in 1997, this famous transgenic cow completely shattered the whole expectation that biotech strictly belongs to laboratory mice. Now, what's really interesting about this slide is seeing exactly what made Rosie's so revolutionary: her milk. Unlike standard cow's milk, Rosie's was specifically engineered to be enriched with a human protein called alpha-lactobumin. This was a huge deal because it addressed a critical nutritional need, offering a far more nutritious option for human infants. Basically, Rosie functioned as a living biological factory, churning out this highly valuable agent just by doing, well, what cows do naturally. All right, moving into our final area, Section 4, testing safety and toxicity, and how this protects public health.
So, the crucial point is that these transgenic models are absolutely revolutionizing how we guarantee vaccine safety. Historically, safety profiles relied heavily on traditional protocols that involved primate testing. But today, transgenic mice offer a much more streamlined evaluation. By using these specifically engineered mice to evaluate vital vaccines, like the polio vaccine, for instance, we've been able to successfully replace a lot of those older primate testing protocols, which significantly accelerates the whole safety evaluation process.
And these benefits extend right into everyday chemical and drug toxicity testing, too. Because transgenic animals can be purposely engineered for increased sensitivity to toxic substances, scientists can efficiently assess chemical effects and evaluate compound risks with profound accuracy. Plus, they can do it in a drastically reduced timeframe. Actually, it's a total game changer for evaluating risk and keeping our medicines and our environment safe. So bringing it all together, whether they're helping us decode basic gene function, modeling complex diseases like Alzheimer's, acting as biological factories for vital human proteins like our friend Rosie the cow, or ensuring the safety of our vaccines, transgenic animals are utterly instrumental to modern biology. They are this versatile, essential living toolkit that is improving human health every single day, which leaves us with a final, provocative thought to consider. As our ability to manipulate DNA continues to advance, how will these architects of life shape the absolute next medical breakthrough? Thank you so much for joining me for today's explainer. I hope this gave you a really clear, focused understanding of the science behind